Sir, you can start now, sir. Thank you.
Okay, so the talk is on structure-based drug design and molecular dynamic simulation. Before going to start talk about the MD simulation, I will say some words about the structure-based drug design approach and where is this computational drug design approach stands in the drug discovery phase. So if you see here briefly, I know that you already know these things quite a lot, and you heard and you saw this type of diagrams many times. But what today we are going to take from this is like a drug development or drug discovery phase. We can divide this as a discovery phase and development phase. So discovery is like initial phase of drug, and then later phases there is preclinical and clinical and formulation aspects. So today's talk is more on the discovery phase, and I will not going to talk on development phase, though computational approaches are still available and they can be used for drug development also. So it is not limited to discovery phase, but in development phase also nowadays these computational approaches like molecular dynamic simulations are used for simulating different nanoparticles or simulating uh, different types of formulations and see the interaction of polymers with the drug and to see the solubility parameter and everything. So these all can be possible using the molecular dynamic simulations. So that aspect is more related to development phase and not on the discovery. So I will not going to cover that thing. But in discovery phase, if you see there is target discovery, hit generation, and lead identification. These are the main points, and then the lead optimization, of course. So the selection phase here is very crucial, and this is a basic research area where normally the computational scientist works. So if we briefly describe this, it can start from anywhere. So it's not like there is no clear-cut starting point for this. So it may be like you already have a structure of the protein with a ligand, and then you start designing the drug molecules. Or maybe sometimes it may be possible that you started synthesizing the molecules without having any design approach. So you may or may not have the design thing in your research plan, or you may start with you have started with the organic synthesis. This is a conventional approach, we can say. Like in conventional there is synthesis, and then we test the compounds using some in vitro assay, and then we go to the preclinical and then clinical research. And in between that, we predict the structure activity relationship of the compounds based upon the activity data we receive and the structural information we have. And then there is one more approach which is today these days are used is like rather than synthesizing the organic compounds, there are number of commercial libraries of compounds are available. And these commercial compounds can be screened using the high throughput screening process, where the amount of there are like number of targets are available, number of kits are available. So using that target kits, specific activity of compounds is checked. Like if we say, like if you want to see antimicrobial activity, what we do is minimum inhibitory concentration we find out, but it doesn't give us a very clear-cut idea about the target which we are searching for. So to get more detailed information of the drug target, so we can have a specific enzyme, and then we can check whether that enzyme is inhibited by that particular drug. So that approach can be included by using this high throughput screening process. And then if the molecule is successful, then it can go to the next phase, like secondary assays that include the assays on the mouse or the zebra fishes, or it may be like guinea pig or something like that, or isolated organs. And then if compounds are found successful, and if these are absorption, distribution, and metabolism properties are found suitable, then that compound IND could be followed, or a new drug approval process could be initiated to make the drug for the clinical use. And then again we come to the same page. Then if we already have a drug in the market, and then we already have some information on that drug, and we can take that information, and then we can again start the phase. So this is a continuous cycle. So, and this is a continuous improvement cycle. So here in this part here, this corner is for the drug design or computational CADD, we can say. And briefly I can say, like, CADD we can divide like that, maybe not clearly. So structure-based drug design approaches and ligand-based drug design approaches. So CADD can be the structure-based or ligand-based. And so if we see the ligand-based approaches, what ligand-based approaches are available? So it may be the pharmacophore. So we already know some information about some drugs which are currently available. Maybe we don't have a structure of the receptor, but we know that particular drugs are active. And then we can have that activity data, and we can compile the information from that ligands to identify a particular pharmacophore which is working, which is giving the activity. Like if we take the example of steroid nuclease, so you know that we already know that steroid nuclease is very specific on the steroid receptor, though the steroid receptors are not identified till very clearly. So in that case, we can have the ligand-based approaches for drug design. Then we can have the 3D QSAR, where we, instead of just seeing the pharmacophore in two-dimensional view, we can see the structure of the compounds in three-dimensional view. Now, the basic difference between the normal QSAR and 3D QSAR is about the conformations. So in 3D QSAR, you can have a number of conformations of the same ligand. But in a normal QSAR, we just have one view of that particular ligand, and we don't know much about the different conformations which are possible for that particular compound. So it gives more detailed information if we go for the 3D QSAR. Then there is also a similarity approach, scaffold hopping. Like it might be like the pharmacophore is not matching, but it might be possible that the scaffold is matching. Like if you see this diethyl stilbestrol derivatives and then steroid nuclease, they doesn't match clearly. There is no clear-cut pharmacophore. But if we place the stilbestrol derivatives over the steroid nuclease, they match. So that is called the hopping techniques. And then we can also predict the absorption, distribution, and metabolism property. But this is all the basic things. We using these approaches, we can come to know about that certain ligands could bind or certain ligands could not be bind. But to get more detailed information, whether these compounds are binding, or whether these compounds are showing activity, or how they are interacting with the receptor, we don't get that information. And for that, we need a structure-based drug design approach. Now, for the basic requirement in the structure-based drug design is we should know the structure of the ligand and the structure of the receptor. So ligands, we already have the chemicals which are synthesized, but the real limitation is with the receptor. So previously, the receptor information was very scarce, and there are very few receptors who are available. But nowadays, if you see, I just want to show one recent view on the currently available resources on protein data bank. So if you see here, the statistics, current statistic, this is today's statistic. And if you see the protein data bank, the number of structures, can you see this? Can you see this window?
Yes, sir. Yes, sir.
Okay. So I will try to switch back and forth between these slides and the browser also. So if you see the number of entries here, this is the year. So it all started from 1976, and if you see the number of entries from 1976 to 2021. So currently there are 51,53,825 targets are available, and each year there is addition of 1,000 new targets. So each year 1,000 new structure of the proteins are deposited in protein data bank. This is only from the X-ray crystallography, and not this is only the data. I think it's for the X-ray crystallography data. And if we go for the The other methods like NMR method, because we can also have structural information from NMR. So nowadays the NMR is not that very core in the structure identification, but there are some certain structures which are identified by NMR techniques. So currently there are 13,270 structures are available through NMR. And the new technique, which is very emerging now, is a cryo-EM or cryo-electron microscopy technique. And using that techniques, if you see, there is a tremendous increase in the structural data available from 2015 onwards. So there is an exponential growth of the structural data availability using the cryo-EM techniques, and almost 6,911 structures are now deposited in protein data bank. So this protein data bank has the collection of all these structures which we require for structure-based drug design. But sometimes it may happen that you don't have the just a second. Sometimes it may happen that you don't have the structural data available through these techniques, and then in that case you may need homology modeling. So homology modeling is the technique where we have the structure of structure of the protein which is related to the other protein, but it is but the identity is not identity may be like 99%, or it may be like not 99%, but it's like 90% or 80% or 70%. And then in that case we can model the receptor structure based upon the structural data which is available for the other receptor, so or a close relative of that structure. So to do the homology modeling, we need to identify the sequence of the target protein, and then we compare the sequence of the structure which is available, and then we build a model based upon the structural data available. But the real limitation for homology modeling is about the similarity. So how much similarity is there between the two sequences? So if the I can say, like, if the sequence is more than 60%, then you can build a good homology model from that. But if you don't have the similarity beyond if it is like less than 60%, or sometimes it may be like less than 40%, then it's very difficult to build the homology model for that. So you must need the structure in that case, and you cannot proceed with that structure if the structure is not very efficient. Though there is ab initio approaches are also there for homology modeling, but nowadays they are not so much used as compared to the real structural data which is available in protein data bank. Then I have prepared some sort of questions like what we are looking for, what are the targets, and what level of understanding we got from that. So it's like these are the questions we must ask ourselves when we start any research. So we should ask about what we are looking for. So what is we are looking is like which protein you want to you want to study, and then and what is the target of that protein, where the protein is located. Sometimes the protein is located on the cell surface, so it may be a membrane bound protein, or it may be a cytoplasmic, or it may be nuclear protein. So expression level and the availability is and the post transcriptional modifications are very critical for the choosing the target. So we must know whether where the target is located, and how we are going to bind that target. So if the target is on the cell surface, then it may be like you don't need the more lipophilic drugs. Or if the target is located inside the cell, then you must cross the cell barrier to reach to the target. So we must think about in that way. And also in the whether the target is the bacteria, or is a virus, is it a human host protein. Depend on that the selection, and then the next things will depend, like organisms could be human, bacteria, or virus. Nature of target, just I say like, or it may be sometimes it's most of the times the target is protein, but it might be a small peptide, or it may be a DNA, or it may be RNA, or it may be a specific enzyme you want to target. So that information you can search from the BLAST. So you know BLAST. So the BLAST is the database which is available for the structural information. So this is nucleotide BLAST. So if you want to search for the sequences, protein sequences available, and if your target is DNA or RNA, then you can go for the nucleotide BLAST. Or if you have the protein you want to study, you can go for the protein BLAST. So when you go into BLAST search, you need to put the FASTA sequence or accession number for that sequence, and then you can search for the related compounds, or it may be you can choose the database for that. So if you want to search for whether which receptors are available, or which protein receptors, 3D receptors are available, then you can modify the search. Like here you can have this app, like you want to search that particular target in protein data bank. So you will get a list of the uh, receptors which are available in protein data bank for that particular sequence. So you need that FASTA sequence for that purpose. Uh, then okay, then as I already told you, the location is whether the protein or the protein is located extracellular, intracellular, or cytoplasm or nucleus. Then the availability of protein 3D structure. So if the structure is available, then good. That you don't have to model the protein. And if it is not available, then you need to model the protein. Then I think I will go all these questions after this slide maybe. And then and if you have the 3D structure, then what to do next? So you need to choose the structure, right structure, and then you need to choose the software package. Like there are various freely available softwares, or academic versions of softwares are like available. Like Autodoc Vina is completely free, but you can also search for OpenEye or RDKit softwares which are available, and they have the collection of this new tools that you can use for docking and next phases of research. Then it is important to process how we are going to process the structure, how to find the binding site, because it's not only the choosing a structure and then start docking anywhere. So we must ensure that you find the right site, right mechanism, and then you are targeting that site. Or it may be possible that there is no information available on the binding site, then you need to find out the binding site by your own or from the literature. Then you once you find out the binding site, then you can build the grid around that binding site, and you can perform docking. Uh, then once you complete the docking, then you can visualize the poses of your compounds, dock poses, various dock poses. And then it's not only a docking, because it's a one approach to make the drug available with the receptor, but it is not finished. So it's just a starting of your research in computational research. And then you must ascertain what is the binding free energy available for that, and it is very important. And then, because we know that the protein is not a rigid structure, and physiologically, if we compare the protein in the body and protein in the computer, so we must have a situation where our protein is present with the water and not without the water, because it is impossible to imagine the protein without water, because our body has 70% of water, and then every receptor is submerged in the water. So there are a number of interactions which are depend on the water, and so we must have a model which is which corresponds with the actual physiological model of our body. And is the docking is enough? So these questions can arise. So I will I will try to answer these questions. These questions I asked for myself, and then I'm going to define how to defend these questions, or how can we answer these questions. So first we need to make sure that what is FASTA coding. So I just mentioned about that. If you want to search for the structure, then there is a FASTA coding. So this is FASTA, standard FASTA code for the amino acids and different uh, nucleotides present in RNA and DNA. So, and then also the codon sequences, how they call. So normally the amino acids are labeled as per the names, but sometimes it may be possible that say, the amino acid have the same starting name. So like arginine is not called R, but it's arginine. So it starts from ARG, or the small form is like ARG. Isoleucine is, the I is there, so it's like it starts from isoleucine or ILE. Methanoin, and then histidine, uh, and glutamine, proline. So this is a standard FASTA coding, and then amino acids are called like that only. And then also about the codon sequences, if they're there, then STP stands for stop codon, or STP, this stop codon, or this is about the amino acid coding. And then the 3D structure which is available in the PDB has different information. So what information it has? So the first is the atom type. So it may be the atoms of the amino acid. So it is labeled as atom entry, or sometimes if the ligand is bound, then its entry is like hit atom and not the atom. So hit atom stands for the heterogeneous atom. So it's not the amino acid. So that's why it's called hit atom. And then the next row is for the atom numbers. So atom number row, and then it is followed by atom type. So if you see here, like this is a backbone atoms of the of, of the amino acids. So C beta, C, and then C alpha, like that, and then terminal residues. Then amino acid type. So here I just mentioned about the coding. So here the leucine is coded as LEU or arginine or ASN, something like that. And then next is the chain. So sometimes you may have a receptor with a number of chains. So it may be chain A, chain B, chain C, or D. So sometimes the chains may be identical, or it may be a different one. So that information is included here. And then the residue number, like here the leucine comes at 151 number, and then the this is at 149, or one arginine at 149 or 150, something like that. So this is the residue information. And then the next crucial information is XYZ coordinates. So where these atoms, individual atoms of that particular amino acid of that particular protein are located in 3-dimensional space. So this is the coordinate file. So, and that's why PDB file is also called as a coordinate file. So these are called as initial coordinates. So these are the coordinates which obtain, which we get from the X-ray crystallography or cryo-EM technique or NMR technique. So NMR or cryo-EM uh, or X-ray crystallography techniques gives us the information about the protein in these terms, and then we got the initial information about the coordinates of that particular residues located all over that protein. So once we got this, then we start with the next phase of drug discovery, that is molecular dynamics simulation. But I think before I go, I'm going to show you what is molecular dynamics simulation. I just want to inform you about what is docking and what docking is. So if you see here, can you see the receptor, or is it not visible properly? Can you see that? Or is that okay? White background or black background?
Yes, sir.
Yeah, now it is clear, sir. Visible.
Okay. So I will make it the white. So if you see here, the cyan color is a ligand, one ligand there, and there is one protein. So this protein is present in the as a ribbon like structures, and then ball and stick arrangement of this ligand is there. So this is one of the dock pose. So I did docking of one of my project, and I did this docking. Now if you see the now how I have performed docking, and I think this docking topic is quite largely discussed in the previous presentations also. So I am not going to go into the brief about that, but just want to inform you about that. When you dock the ligand in that particular receptor binding cavity, so this is the receptor binding cavity, and then if you see the different orientation, so the ligand can adopt different conformations, and this conformation depends upon the flexibility which the ligand has. And also this it's not only the flexibility, but it also under the limitations of force field, because we know that the carbon-carbon bond has particular angle and particular distance, and the force fields are the parameters which parameterize these distances between the atoms, and that's why it is not possible that you will get a very unusual pose. So unusual means you cannot bend the angle up to certain degrees, and there should be some limitations. So these limitations and these a standard scale is set by force field. So force fields are the basic parameters which governs the small molecules as well as the big protein. So if you see here, I just make the structure like that, and then if I click somewhere, so if you see that there is different orientation of the same ligand, and then the orientation has changed. Now it has changed different orientation. So these are different conformations. So same ligand bind to the same receptor in different orientations. So these orientations largely depends upon the site which is used for the docking, and also on the flexibility of ligand and how efficiently it accommodates. So depend upon the this docking program, you can have the different outputs. Maybe you can set different outputs of dock poses, different conformations. Now here you can see that when I'm moving, when I'm switching to the different ligand only the ligand conformation has changed, and there is no change in other part of the protein. So protein is held rigid, and ligand is flexible. So this is yesterday in a Dipti Madam session, you may heard about this, the what we say, the rigid docking and flexible docking. So what is different? So here in rigid docking, the ligand is not flexible, so it will dock, but that is not a very good approach. So rigid docking is not much followed now, but it may be needed for certain reasons. It might be, and then it can be the point. But in normal circumstances, we have a rigid receptor, and then we dock a flexible ligand there, and then we get number of poses, and we compare the poses based upon the docking score. And then as we don't have a flexibility here, because you see that this ligand can adopt like 1, 2, 3, 4, 5 different conformations. Now imagine this small ligand adopts 5 different conformations. So how many conformations could be possible for this big protein which we are taking for the binding? Can you imagine how many conformations will be there? Yes, anyone? So there will be infinite conformations, and we don't know which conformation is actual conformation. So how to proceed with that? So this is very challenging one, because and you cannot do that using docking. So if protein is adopting like 1,000 conformations, you cannot take one ligand and dock in a different conformations on that 1,000 different proteins. So it may take maybe years to complete that process. And so this you cannot do that. And one more thing, the docking is a process which makes the ligand available in the binding site. And it is not like the process which is normally followed, because in normal physiology, if the protein is somewhere in the body, then ligand should go and then bind to the bind to that protein. So this is what a physiological process. But what we do is we apply some bias, and then we pose that ligand into the binding cavity. So of course there is some bias we use because we need to dock that ligand into the protein. Now you may ask, like, why we are not allowing the ligand to be somewhere here and then allowing the ligand to go and bind to that side. Yes, it is possible, but it may take some years to do that process because the processes which is taking into the macromolecules, the bond length change and bond angle change process, they are very like 10 raised to minus 15 seconds. They are taking place at 10 raised to minus 15 seconds, means you need to calculate that process in femtoseconds. And in that way, it may take years to complete this process using the computational approaches. And that's why we are applying some bias to do, and then we do the docking. So we are not doing the process where the ligand coming and then it going and inside the binding pocket and then it is interacting. But instead, what we are doing is we are cutting off this process because it is going to take a lot of time, and which is not feasible at this moment. I think it's not possible at this moment. And that's why we do the docking. So docking is a simple one step which gives us next to the next phase of the structure-based drug design approach. So once we got a docking, so it's very difficult to choose the poses, because if you see, I just want to show you the result of this docking. So if you see here, so these ligands are binding with this docking score. So if you see, the docking score of this ligand is -5. The docking score of the second pose. So same ligand binding with the different poses. So one pose is -5.8, the other is -4, the other one is -3, the -3 and -3. Now, if you just imagine that this is the top ranking pose, and then I will go with this pose, then it may give some wrong prediction, because we are not doing this in presence of water, because there is no water in the receptor. So what we can do is we can apply some binding energy prediction approaches, like in Maestro we use the prime based approach, where MMGBSA method is used. MMGBSA is a method where we imagine imagine that there is a program which imagines the water molecules around the binding site, and then it calculates the favorable and unfavorable interaction with that water molecules. And these water molecules are basically the mathematical model of water, and not exact the real model of water. So it's not like the real model, but it's a mathematical model. And using that mathematical model, we predict that which pose is having high binding affinity. So if we compare here, now in docking, the maximum I got with this pose, but now that pose is having a score -40.15. So its score has been reduced as compared to other poses. And here I come to know that this is the pose which is having a very good binding affinity as compared to these pose. So MMGBSA based approach gives us more detailed information rather than just a docking score, where we don't imagine any water there in the binding site. And hence the sampling is more efficient when you have this binding energy prediction approaches. But still, it's not the confirm that this is the pose which is having the interaction with the receptor. So what we can do is, or what we do is we take this pose with the receptor, and then we perform MD simulation. Now the question is, what is MD simulation then? So if you see the basics of molecular dynamics simulation, then you can understand that particles are in MD simulation, particles are submitted to interatomic potential, which is, we can say, like U, and this and which is based upon Newton's law. And this interatomic potential is expressed in terms of classical mechanics, or it may be expressed in terms of quantum mechanics. So if we express this in the quantum mechanics, where we use the Schrodinger equation, then that approach of MD simulation is called ab initio MD simulations. So quantum mechanical approach are ab initio MDs, while in classical mechanics, which is called as a classical MD, here the expression of U is in terms of force field. So we already parameterized some force field from quantum mechanics, and then that force field which is generated from quantum mechanics are used as a reference for designing the new molecules for classical mechanics. So in this term, if we see the accuracy of both methods, then quantum mechanics is obviously very accurate, but the real limitation of quantum mechanics is the time. And to calculate particular ligand interaction, or even for a small molecule, it takes 1 day, or even on the supercomputer, it takes 1 or 2 days to complete the quantum mechanical calculations. So if you imagine the protein, then it is in the current situation, I think it's completely impossible to do quantum mechanical calculation for a protein. That could be a crazy idea then. So in that case, we use the force field rather than using the quantum mechanical approach. We use the force field to do molecular dynamics. So the idea of this molecular dynamic simulation is that all atoms are moving, and then if you calculate the forces between the atoms at the function of time. So suppose one atom is moving at 1 second, at the first second, and then the moment after a certain moment, the atom is at different position. And then if you calculate the position of that atom at the different times scale, then we can get the expression like that, and which can be summarized in terms of Newton's law, as F is equal to ma. And if you try to solve this equation by using the Newton's law, then we come to know about this one. So this is what, and here we will stop, because it's not only one atom we are considering, but and one atom has 3n degree of freedom. So if you imagine the protein, it is having thousands of atoms, and then you have water, and then you have protein, and you have your ligand there. So if you want to simulate that big system, it is impossible to calculate that forces in one single go. So you cannot use this equation directly to calculate that forces. It's impossible. So there is a possibility that if you calculate these forces which is exerted on atoms in different time period. So if you measure the forces and in the n number of time period, so it can be possible. So arithmetic solution is possible for that approach. So what we do is, suppose this atom is moving. So you can see, like, suppose this is the protein which is present, and then if the atom is moving, so one atom comes and then its movement started. So every atom is moving, and then the velocities of each and every atom is calculated at that particular time frame. So in every time frame, we calculate the distance moved from the original position. So we have initial coordinates in a protein data bank we take, and then we bind the ligand to that protein, and then we calculate in a one step, like suppose we take 10 raised to minus 15. So it's like 0.001 picosecond. We measure the coordinates of that protein, and then as the atoms are moving, so what will happen that on the next picosecond, what will happen that coordinates will change. So initial coordinates will change to a new coordinates. Now protein will orient in the new coordinates, and then we calculate the forces on that coordinates, and then this process will be repeated until we got the minima. Because if you want to calculate the forces the protein structure which is deposited in protein data bank is not energy minimized structure, and we don't know what is the energy minimized structure is there, or which conformation is energy minimized. So that information we don't have that. And so what we do is we apply the force, initial force, and then we start to minimize the protein. So when the minimization started, so it orients in a different conformation. Sometimes it may go to the saddle point, or it may go to the minima, and then there is a conformational barrier. So here what happens that once, if we stuck into this barrier, so if the protein structure is minimized at certain extent, then what will happen? We need to increase that minimization, and we need to apply some force to bring back that conformation to this minima to the saddle point, because the real minima may be somewhere here and not here. So when you simulate that for a long period of time, so like suppose if you simulate this protein for 10 nanoseconds, so what will happen? Like in 10 nanoseconds, maybe your protein comes to this minima, and then you assume that your conformation reached to the minima, and then you calculate the energies. But this is not a real conformation of that protein. So what will happen that if you break this barrier, so if you extend the simulation for a longer period of time, like 100 nanoseconds or 1000 nanoseconds or 1 microsecond, or so then what will happen that your structure may cross the barrier and it will go to the real minima. So this is very critical point in MD simulation, that your conformation reaches to the minima. And once your structure, and how to check that? You can check using the RMSD. So once the RMSD is stabilized, so you get the idea that your conformation reached to the minima, and then whatever interactions are taking place between the drug and ligand you can calculate that interactions, and you can calculate the energies, because energy of the protein is now minimized. So there is no change happening with the protein. Your ligand energy is minimized, so both are minimized. So when the ligand energy is minimized and then protein energy is minimized, so there is no chance of fluctuation in these. So whatever change will happen is due to the binding process, drug binding to the receptor, and that's the idea of molecular dynamic simulation. So if you see the forces, so if we imagine the forces in terms of quantum mechanics, then the changes happening can be recorded in terms of femtoseconds, picoseconds, up to nanoseconds. So quantum mechanical calculations are really fast as compared to classical mechanics. But to calculate these forces, we need a very, very highly intense computational infrastructure. And I think the quantum computers can solve that. Maybe they can solve this, but still the quantum computing is still not. I think it will take some time to build the quantum computer. So what we do is a classical. So mostly what we do is a classical mechanics. In classical mechanics, the changes happening inside the molecule are ranged from nanoseconds to few microseconds to milliseconds to seconds. So changes happening in the protein are in terms of nanoseconds to microseconds to milliseconds to seconds. And so to capture all these changes, we need to simulate the protein as long as possible. So currently, if you see the literature, then previously the simulations were in picoseconds. Then in 2000, it is reached to nanoseconds. This is with respect to the advancement of computational power, and with the supercomputer facilities, the simulations can now be extended to up to microseconds to milliseconds. So now the millisecond simulations are now possible, and the next goal is to reach this simulation to the second scale. So this is really a big task to make the simulation from nanoseconds to microseconds to milliseconds to seconds, because you are dividing the simulation. This is not a real clock we are imagining. So this is the computational clock. So you are taking the trajectory, which is recording the trajectory, which is in the femtoseconds. So currently, if I have a protein of 500 amino acids, imagine a protein with 500 amino acids, and then if it is submerged in the water and it has suppose one ligand bind to that protein, then on a current supercomputer cluster which I'm having, it takes like around 12 hours to 24 hours to complete the simulation for 100 nanosecond. So imagine the same thing for the microsecond. So how much time it will take, and just imagine how much time it will take for milliseconds and seconds. So, and I'm not talking about the normal computer which we use for that. So that could be in the range of picoseconds. So it may take a month to complete a few nanosecond simulation on normal computer. Then to have that simulation, we need to, it's not like you just take a protein and then you start simulating, or you just take a protein and then you have ligand bound to that protein and then you are simulating. So simulation has to be proceeded by a number of steps. So you need to have a choose of solvation. So now this solvation is a real solvation model and not the mathematical model. So previously I talked about mathematical models like MMGBSA or PBSA solvents models. Now these real models of solvations are like defined in terms of periodic boundary conditions. So here what happens is, like, when you simulate this center box is your protein, and it is imagined that your computationally it is calculated that your protein is surrounded by the same replica of that. So what happens is, like, if though the atoms are moving like that, so it start appearing from the back. So if atoms are moving like this, so it will start again appearing from the back. So the protein cannot see its image, and if it is going to see that image, then the simulation will not work. So we need to build a periodic boundary condition around, and then you can use the water surrounding that protein. So you can submerge the protein into the water, the whole protein or part of the protein. If you do that part of the protein, then it's called a stochastic boundary condition. But normally the PBC is the approach which is normally followed. Then there are some implicit solvation models are also there, which are called the mathematical models. So this is the famous Poisson-Boltzmann equation, and this is called PBSA solvation model. And this is another equation, that is a generalized bond equation, which deals with the solvation energy. So this is GBSA solvent. So these are currently two most used solvation models available for calculating the binding free energies in presence of water. And then once we submerge the protein into that protein bound ligand into the water, and then we need to apply some ensembles. What we need to do is selection of interaction model, then selection of boundary condition, which we see in the last slide, then selection of initial conditions. So these are the initial velocities, like PDB structure we need, so which is already bound to the ligand now. So that velocities we need, and then we need to select the ensemble. Now ensembles are like is a condition where we are simulating. So it's a normal temperature pressure condition. So where there is no pressure boundary condition, or there is no change in the pressure and temperature, there is infinite change with it is open system, where open to the surrounding system, NVE or NVT, where the pressure is fixed but temperature is not fixed, or NPT, where temperature and pressure both are fixed. So there is infinite source of pressure and temperature. So if temperature increase or decrease, so it can be equilibrated with that ensemble. Then selection of target temperature, what temperature you need simulate. Normally the simulation is proceeded at 300 degrees Celsius. Then the pressure condition, which is normal atmospheric pressure. Then we need to choose the integrator, like normally the integrator and thermostat and barostat. So these are the mathematical methods which are available, which serve as an integrator, thermostat and barostat, like Langevin thermostat or Nusshofer or Bernstein thermostats are the famous models available. Then we need to perform simulation until equilibrium is reached. This is very important, because normally when you start simulating the protein, then protein has some strain inside. So if you directly start the MD simulation, production, the model could be collapsed. So to avoid that, we need to minimize the energy of the initial coordinates. So we first minimize the model by applying the small forces, and we apply the restraints on particular some atoms. And then when simulation start proceeding, then we relieve that restraints from the protein, and then we start actual simulation. So the simulation is always in the phases. So there is a minimization phase, then there is a equilibrium phase. Once the minimization equilibrium is reached properly, then you can do the production simulation. And then in production simulation, we can choose like how long we want to simulate. And then once the simulation is completed, then we can analyze the trajectories which are generated from the simulation using different trajectory analysis tools. So what information we got from this simulation, that we get the equilibrium structure of the macromolecules, because in docking we don't have the equilibrium structure, it's just a rigid structure. So in simulation we get equilibrium structure of the macromolecules. Then we can also calculate the fluid density and at different pressure and temperature condition, which is not happening with the docking model. And then we can calculate the radial distribution functions, and how the movement happening inside the inside that protein. And then we can also calculate the energetic data like binding affinity, what affinity the ligand is having, and how much long the affinity is there. Like if you simulate the ligand for a longer duration of time, like 100 nanoseconds or 500 nanoseconds, then it may be possible that initially you got a very good binding pose and your ligand is stay there. And when you start simulating, then gradually that molecule is getting out from the binding site, because it is not having a stable interaction with that binding site. So it will gradually come out, or it may flip its position, or sometimes it may be possible that the dock pose is not exactly the pose of that ligand, and so it will change the pose in between that. So this all information we can get from the simulation. Then we can calculate the heat capacity, thermoelastic coefficients. Then nowadays these molecular dynamic simulations are also used for calculating the diffusion coefficients, thermal conductivity, and viscosity, and many more applications. Currently, if you see the different softwares and the force fields which are available, so the CHARM, then Umber and GROMAX, GROMOS and NAMD. So basically, Umber software tool or GROMAX software tools are available, or NAMD software tools, or Desmond. I think Desmond is not in the, here. I didn't place in the list, but Desmond is another software which is now integrated with the Maestro, Schrodinger Maestro. So Desmond can be used, or Umber can be used, or GROMAX or NAMD. In that GROMAX and NAMD, I think they are freely available. So you can use that tools for the simulation. Umber is a proprietary, so you need to buy that package for performing the MD simulation in Umber. Or even Desmond, you need to buy the software. And then there are different force fields which are available, like CHARM force field. Previously the CHARM 27 force field was there. Now CHARM 36 force field is available. Then Umber force field, which is quite famous. Then GROMAX has its own force field, but now even GROMAX uses the Umber force field. Then Desmond, which is having its own OPLS-3 force field. Here are our resources which are available for performing all these type of simulations we normally perform. We have currently a Puthi supercomputer, which is having a speed of 1.3 petaflops, and then we have another supercomputer called Mahati, which is having which is ranked 48th in the latest supercomputer top 500 supercomputers all over the world. And then the Finland is also going to launch this new supercomputer called Lumi. I will just introduce a small video is there for the Lumi supercomputer. So it's called Euro HPC. So this is available for all over the Europe.
To host one of the Euro HPC world-class supercomputers. A group of countries participates in this unique joint endeavor called Lumi to acquire, build, and deploy a world-class computing and data infrastructure. The supercomputer will be placed in CSE's data center in Kajaani, Finland. When the operations start in mid-2021, it will be one of the most competitive supercomputers in the world and a leadership class platform for artificial intelligence, providing HPC and AI resources for European researchers in academia and industry. The LUMI supercomputer's peak performance is an astounding 553 petaflop per second.
Now, if you see the speed of this, so this computer is going to have a speed of 550 petaflops. Now, currently the world's top supercomputer, which is Fujitsu in Japan, is having around 200 petaflops speed. So it might be the double of that.
The computing power of 1.5 million modern laptops. This many laptops would create a 23.25 kilometer high tower. Its storage system consists of 3 layers. LUMI F, a 7 petabyte flash technology based storage tier, 80 petabyte Lusta parallel file system, and LUMI O, a 30 petabyte object storage service. The LUMI system is an HPE Cray machine supplied by Hewlett-Packard Enterprise, HPE.
10 European countries have invested together with EU to a single environment, joint systems, and placed it in the one of the most eco-efficient data centers in the world in Kajaani in Finland. So this
a considerable impact, as supercomputers consume plenty of electricity. CSE's data center in Kajaani has an abundant supply of low-price and environmentally friendly hydroelectric power. The location's benefits include warm water cooling, which enables waste heat to be utilized in Kajaani's district heating network, further reducing costs and CO2 footprint. The CSE Kajaani data center is highly scalable for large hardware installations and extensions or other potential infrastructure, making it ideal for building a sustainable data center ecosystem. The LUMI supercomputing and data infrastructure at CSE's data center in Kajaani will help position Europe as one of the world leaders in supercomputing. The computing power of this size is required in leading-edge research in a wide range of data and computing-intensive fields, as the importance of various data-driven methods in research, administration, and industry are constantly increasing. LUMI will also have a channel for urgent computing. This type of allocation will allow us to grant some of LUMI's resources on an ad hoc basis for time and mission-critical simulations. With the new infrastructure, European researchers can access world-class computing resources, which have a direct positive impact on European research in nearly all scientific disciplines. The state-of-the-art computing resources will also lay grounds for carrying out research in areas that have previously been out of reach, increasing the possibilities for scientific breakthroughs with immense societal impact, such as understanding climate change. To summarize it all, the LUMI supercomputing infrastructure will start to operate in mid-2021, benefiting European innovation and research.
So this is about the LUMI supercomputer. LUMI stands for snow in Finnish language. And this is our typical crystallization lab. So I just mentioned about the X-ray crystallography. So for to get information about the 3D structure of the protein, we need to have a protein. So normally for X-ray crystallography, if you want to have a protein structure, it must be about for a few micrograms to a few milligrams amount of quantity of the protein is required. And this quantity should be very pure. So the purity is almost like more than 95% or more than 95%. So for that purpose, the protein need to be purified. So this is a typical AICTE purifier, which is used for protein purification. Then once the protein is purified, then the thermal stability of the protein is checked so that the storage condition and screening techniques can be optimized for that particular protein. So this is nano DSF, nano DSF, which is used for thermal stability of the protein. Once the thermal stability and protein purification is over, then protein is seeded into the wells. So this is a typical 96 well format of the wells. So this is called crystallization wells. So this is a typical well. So if you see, this is a sitting drop method where the protein drop is applied using, it is not possible to apply these drops using by hand. So this mosquito machine is used to apply the protein into a few in a few microgram microliter quantity into each well, and then each well is seeded for few days or few or maybe months to achieve the crystallization. So the wells, as we are having a number of wells, and each well is, it may be possible that there may be crystal growth in particular well and there is no crystal growth in other wells. So to monitor this well growth of the crystal, the this every screen is then placed into this formulatrix rack imager. So in rack imager, what happens is like there is a robotic arm is there in the back side. So it takes the this plate and then takes the photographs of this like this. So at a particular interval, so we can set interval for days like 5 days and then 10 days or something like that. So each of that day the plate is taken by the robot, and then it scan the whole plate and takes the photographs. Once the photographs are taken, then they are included into this, and then we can visualize the photographs. Once we confirm that there is some crystal growth, then we have this PX scanner, which is used for taking the X-ray image of that protein, just to confirm that whether the crystals are of the protein, or it may be sometimes possible that buffer may crystallize out. So we need to confirm that protein is crystallized and not the buffer or any other material which is present inside the well. That can be confirmed by this X-ray machine. And then once we confirm that we have a protein and we got the crystals of the protein, then the sample is placed, is then sent, or we can bring the sample to this synchrotron radiation sources. These are the highest energy X-ray which are available. And this here the X-rays are revolve around at a very high speed. So these machines never stop, so they are continuously working. And in this, the X-rays which are it's just like one football ground. So in that, this is one of the synchrotron source which is placed into Grenoble, France. And there are many synchrotron sources are available in Europe. There are around, I think, 7 to 8 different synchrotron sources are available. Here we can take our protein, and then the crystal images are recorded for very high intense, using very high intense X-ray beam. And then after taking that images, then images are solved by different software tools. Again, there is use of molecular dynamic simulations to perform and then ascertain the images. And then once we got electron density maps, then the maps is fitted into the sequence, which is sequence data we have for that particular protein. And then the 3D coordinates of the protein are deduced from that information. And then once we deduce the information, then it is deposited in protein data bank. So this is what the process of doing X-ray crystallography or protein purification or protein production, then purification, then crystallization, and then recording the images and then depositing into the protein data bank. And then the researchers can use that 3D structure for docking, and then for again the cycles again repeated. So this is our goal. This is our group, and this is my supervisor, Professor Autisalu Ahen, and these are our group members. And I'm thankful for Abo Academy, then Academy of Finland, Torjo and Pentti both Memorial Foundation, Biocenter Finland, and CSC Resources for doing these all these activities as a postdoctoral researcher here in Finland. And I am thankful for Purna Kirti Chandra for giving me the opportunity to talk on this AICTE sponsored workshop. Thank you.
Thank you, sir. Thank you for giving a special lecture regarding the modern technologies in drug discovery, sir. Thank you very much, sir. Participants, if you had any queries, you may ask sir directly now. So that sir said so many new technology things, that means recent technology things that are going to be happen in the abroad. So whatever the doubt you are having, please ask sir. So you best utilize the chance now. So participants, if you had any doubt. Please raise your hand. Yeah. Okay. Sir, Purna sir, you can ask.
Okay. Hi, Dr. Rajendra.
Yes.
Yeah, right. How far docking score is important in getting our intended pharmacological activity or an unintended toxic activity for a molecule?
I think docking score is not that important for activity related information, I think, because docking score is just for the initial step where we want to choose particular ligands for the further process like MD simulation or something like that, because with just docking we cannot confirm that this ligand is going to bind. So